Thank you everyone for joining today. I am going to run a workshop on biophysical muscle models. I will start by giving a brief overview of the muscle model, and we will have a chance to play with the code actually. So here is the sign-in link if you didn't do it yet. All right, let's get started. It may sound really obvious, but sometimes I find it a little bit surprising to think about all the muscles that are generating movement. So when we move, basically everything comes from the muscle. The muscles are pulling our bones and moving our bodies. So about modeling the muscles, I'd like to see how people are familiar with the muscle models. There's the reaction button on the Zoom. So can you indicate whether you heard about heel-type muscle model by pulling thumbs up? And if you heard about close bridges, the clap. And then if you are not familiar with either of those, please do the surprise one. Okay, some thumbs up for the heel-type model. How about close bridges? Okay, yeah, a little bit. Uh-huh. Anyone who kind of never heard about the muscle models? All right, yeah. All right, cool. All right, thank you. So these muscle models are crucial for neuromechanical simulation of movements and posture control. And I would say that there are like roughly two ways we use muscle models. One is for doing simulation of movement and balance control, and I'm using it for forward simulation of balance control. So for person on a perturbation platform or an animal, there are muscles that are driving the balance response. And I'd like to study the optimal control strategies or how the control changes when something happens, something changes in your body or in your neural pathways. So I model this kind of an perturbation response using an inverted pendulum model and then put our muscle models and then see what is the optimal feed-forward or feedback strategies. Another way that is commonly used in biomechanics is estimating muscle force from the measurement. So maybe some of you have experience working with OpenSim models. So you can input your measurement of motion capture forces and preferably EMG signal, and then put it into the OpenSim model, and then use the model to estimate how much forces are generated by each of the muscles. So to do these kind of simulations or estimations, we need a good model of a muscle. So how much force the muscle generates given its force or, given its activation or length or velocity. So there are roughly two types of the approaches in estimating muscle force. And I will talk about the conventional or more commonly used way first before moving on to our biophysical muscle model. So approach one is that we can measure muscle forces at various lengths and velocities and use that relationship to calculate the muscle force. It is known that for the muscles, there is an optimal length. So here is the force length curve that we also saw from the previous workshops. And then we will say that muscle force is simply the multiplication of activation, force length, and force velocity curves. And this one we would describe as a phenomenological model, and heel-type muscle model is a type of a phenomenological muscle model. But the problem of using such a model is that it doesn't capture any history-dependent properties, because you can see here, the muscle force is simply the function of activation and its current length and current velocity, and there is no way to capture the history. History meaning how much activation the muscle had before coming to the current stage, or what was the length or velocity before coming to this current stage. But when we look at the actual muscles have quite a lot of history dependence. Here is the conditional stretch of a single muscle fiber. So it is going through lengthening, shortening, lengthening. And then this portion of lengthening must be same as this portion when we go through the same velocity and same length change. But when you look at the force developed by the muscle fiber, here force is different from here. Maybe because not maybe, because the muscle here was coming from the resting state, and the muscle here. Do we see my mouse, by the way? Oh yeah, cool. Sorry. Yeah, so muscle here. Where is it again? Sorry. Yeah, here when it's going through the second stretch, it was already moving before there. So the force it produces is different. But when you plug in the hill-time muscle model, you will end up with the same amount of force. But just a little note that actually Hill reported many of the history-dependent properties of the muscles. So it is not the fault of the Hill, but it is about the Hill-time muscle model that is lacking the history dependence. This kind of a history dependence on the recent activation or recent movement is observed on multiple levels. As I say, it is observed at fiber level, and it is also observed at a muscle level. This is a nice clip from Simon's lab. So if you look here, the muscle is undergoing same type of a length change here. However, these different colors are indicating the different history of the muscles before coming to this part of the cycle. And you can see that the force developed by the muscles are dependent on its history. We also see history dependence on the movement level. Here is the graph from the cerebral palsy participant. So it is a clinical test called knee pendulum test. So when you drop the knee from above and make the leg swing like a pendulum, this is how it swings, the black line, when the leg was held still before the drop. And the orange line is showing how the leg swings if the leg was moved before the drop. So even though they drop from the same height, they will oscillate differently because of the different past. So hill-time muscle model does not capture any of these history dependence, so it is not suitable for some of our projects. So we come up with the different approach of calculating muscle forces based on the muscle contraction mechanism, rather than looking at its overall effect and summarizing it using force length and force velocity curve alone. So we will say muscle force is summation of the close-bridge forces, and we will calculate the probability of attachment and detachment, which I will go into more in details later. And then these close-bridge states are dependent on the past activation and past movement. So by doing so, we will get the history dependence as an emerging property of the muscle model. Is there any questions so far before moving on to the closed-bridge mechanics? I see something on the chat. Let's see. Nope. Okay, no questions so far. All right, let's talk about the physiology a little bit. So active muscle force actually comes from the close-bridge binding between thick filament and thin filament inside the muscle. On the left, we see the structure of the muscle. Muscles are bundled in multiple layers, but deep inside there is myofilament, and there are actin and myosin filaments. And then in between them, there is a molecular binding called close splits. And because the close splits cycles like this and pulls those filaments relative to each other, the muscle produces active force. And there are some passive elements around those myofilaments that also contribute to the overall force of the muscle, but those are passive forces. So active force comes from the close splits and passive force comes from other elements. And we can model that using series elastic element or parallel elastic element on top of the close splits mechanics. So how do we model close splits? We will simplify it and say that close splits is basically a spring that can pull those two filaments. And let's start with the simplest version where there are two states. So they can be either detached or attached like this. And then within the muscle, there are so many of the actin and myosin available. And they are at different lengths, and some of them are attached and some of them are detached. When you make a computational model of the muscle, we will count number of the attached close splits at each length. And then if we have the current number, we can calculate how we will have a transition next step. And that is not a random transition, but there is an probabilistic model on how close splits will transition into another state, right? So here is the attachment rate and detachment rate, and you can see that they are the function of the close split length. So what it says is that when you are near zero, you have a highest probability of being attached. And then when you are pulled away from the resting state, the probability of losing the connection or getting detached increases. So for example, let's say at certain close split lengths, there were 10 detached close splits, and we can come here and read the number on what is the attachment rate, and then multiply them to get how much close splits will get attached next step. And same way, we can look at how much close splits are already attached and read the detachment rate at the specific length and see how much of the attached close splits will lose the connection next step. So overall, we got 90 newly formed close splits next step. And we can do the same calculation for the all available close split lengths, and then calculate the transition of those. Did it make sense so far? Any questions? All right, we'll move on. So we don't do it by hand, but we do it by using a computational model like this. And once we get the distribution of the bound close splits as a function of close split length, we can now calculate the overall force as a summation of the spring forces, which is proportional to the close split length. So the total force will become the summation of each close split force for the bound close split. And let me show you how it evolves over time. So that was quick. So what's going on here is that, all right, I'm trying to stop it a little bit quicker, but all right, yeah. So we were able to calculate the amount of bound close splits at its length and then pull the muscles. So the close splits are physically pulled towards the right. All right, let me see if I can. Sorry, are we still looking at my screen? No? Sorry. All right, how about now? All right, good. So here I'm sorry, you lost it. uh, or like bear with me, sorry, we'll come back. So here, the closed split is at a steady state, and from the attachment rate and detachment rate, we get this much of a baseline attached closed split with this certain distribution. And when we start to pull the muscles, we can see that bound closed split distribution shifts to the rightwards. That is because closed splits that are already attached are physically pulled to the right. So now the number of bound closed splits shifted to the right, but they cannot move infinitely to the right because they will start losing connection because the detachment rate increases as we go further away from the resting state. So if we keep pulling the muscle and increase the muscle length, this is what happens. Now the height of the bound closed splits decreased because closed splits started to lose some connections. And then those closed splits that are detached and filled up can now enter the pool again at the resting length and then start to bind again in the middle. So if we keep pulling and then keep the muscle there, we can see that number of bound closed splits becomes centered at 0 again. Right, we'll go back to the presentation.
We have
Did it?
we have a question in the chat. I wanted to make sure
Okay.
we address that. So I had one question with what technique are you able to find out or look at the number of attached and detached cross bridges?
Are we talking about the experimental methods?
Yes.
Yes. Sorry, I do not know the answer. Is there anyone in the audience who can answer those? I'm more of a computational modeling person.
I think you can't measure it. You fit the data, you adjust the detachment and attachment rates to fit the data. You can't measure it experimentally. This is a really complicated part of assemblages of molecular motors being one of the very toughest biophysical problems.
Mm-hmm. All right, thank you. I don't know, is there a way to look at the molecular procedure actually using some fancy microscopic technique?
You can measure for a single cross bridge using like an optical trap or various other methods like biophysical nanoscale methods. But the problem is then it's only one cross bridge. It isn't an assembly.
I see. Okay. All right, thank you, Kisa. All right, so we are going to test out these force development and how the shortening or lengthening affects number of bound closed splits using the GUI I will provide shortly. So that was the explanation based on the simplest version of the closed splits model, but we can make the model a little more complicated by adding more states. So far we said that closed splits can be either in the attached states or detached states, but there are experimental evidences that are suggesting that there could be other states that can explain the experimental observations. So for example, it is suggested that the myosin can be in a super relaxed state meaning that it is not ready to be bound. And myosin that is ready to be bound can transition into that super relaxed stage by some another rate functions. And also what is important is that it is not that our muscle fibers will just get attached constantly. It is modulated by the control signal that is sent to the muscle. So muscle activation is sent to the muscle fiber as a form of calcium ion. And these calcium ions will enable those actins to be available for the binding. So only after getting the calcium binding, these actin sites become available and becomes ready to produce force. And this is how we control our muscle model in the computational simulations. And this is how much force is produced when you change the concentration of the calcium in the muscle filament in the model. The reason we incorporate these extra states depends pretty much on what kind of phenomena you want to explain using the muscle model. And here we are showing the difference between Bayesian model and two-states model. And this is because our team at Georgia Tech, Emory, and Leuven are interested in reproducing the history-dependent property of the muscle going through this series of stretches. And then we are interested about reproducing these subtle differences in the force development. But depending on your problem, you may not need the three-stage model, or you may need five-stage model, right? So it all depends. All right, so let's test it out using the code I shared. Victoria has already sent out the GitHub page, but if you haven't downloaded yet, please get it from the GitHub. Just letting you know, this is the code we used to provide the tutorial at ISB, International Society for Biomechanics meeting last year, titled Musculoskeletal Simulations with Biophysical Muscle Models, which we did with Lina, Serbi, Tim, and Friedel. And Serbi and Tim contributed a lot for making this possible. So how are people doing with getting the code? Can I have thumbs up if people downloaded the code? All right. Were people able to run it? All right, let's see. Now I will share a different screen so that we can try it out. Here. All right. Did people unzip what they downloaded? All right. Olivia, would you like to sit with me and see? All right. In the code, there are several folders like this, common part 1 and 2. And we are going to go into part 1 and tutorial. Can I get thumbs up if people are here? All right. And let's open GUI crossbridge and hill.m file. All right. And then you can simply run it. It may take some time, but all right. Are we looking at my figure together or is it not shared?
Yeah, we can see your figure.
All right. All right, this is the GUI to play with our closed splits model. Let me tell you some features. So here are the parameters that are specifying the attachment and detachment rates. And I will go into more details shortly. But yeah, that's the control panel to change the attachment and detachment rates. And here is the, oh, you're not seeing it. Sorry. Here is the muscle length over time on the x-axis. And then here is the activation PCA on the y-axis here. And then on the bottom graph, we see how much force is developed from the model. And you can click anywhere here to go through the time and see how much cross splits is formed on the bottom left corner. Do people get it? Right? Does it run on everyone's laptops? All right, now let's simulate shortening. So you can click here on the protocol and then make it to shortening. And then click run. It will tell you to wait a little bit and then generate the force. Is it working for everybody so far? And we can even test the lengthening cycle like this. And as I told you earlier, even though this lengthening looks same as this lengthening, we see different amount of force developed here, between here and here. And does anyone have an explanation of why we have a different amount of force using this GUI? All right, any guess on why the force is different?
Is it the history
Yeah.
that's leading up to it?
Yeah. So let me clarify that we are actually not modeling the history explicitly, but we are just keeping track of how much cross split is found in the muscle. And because when you reach this point at time, that is how much cross split you had in your muscle. But if you went through lengthening and shortening and came to this point in time, there are different amount of cross splits attached in your muscle compared to here and here. And because of that, our muscle model has an history-dependent property as an emerging property, rather than we explicitly kept track of the history of everything. Did it make sense? All right, cool. Now let me show you what we can do with the attachment rate and detachment rate. Well, let me just stay in this window. So here is the attachment rate on the second row of the left column. And F and W's are the parameters we used to characterize this curve. So you can play with it. So if you lower the F, you can see the height of the attachment rate changing. And then by moving W, you can change the width of the attachment curve. And you can see that all the forces disappeared once you changed the property of the muscle. And after setting up the parameters, you can click run again to generate the force using the muscle you just created now. And we can see changing forces here. And here comes our break room activity. Give me a second. Where's my PowerPoint? Right. Oh no, my PowerPoint wants to crash. Just give me a second. Any questions, anyone? While I restart my PowerPoint.
When you change F and W, does it change like the history of the I forgot what the like little graph was called, but does it change the history of that as well?
No, it wouldn't. Or we are going to simulate it from the beginning. So when it reaches the steady state, depending on your muscle properties, your muscle may have a different amount of cross bridge attached at the resting state, but not that we are actually keeping it in the history. So,
Okay, that makes sense.
right.
We've got another question here in the chat. Amelia asks, so the main history that this model is tracking is the amount of attachments that it had at the start. Is that correct?
Yes.
Okay.
Well,
Thank you.
all right, thank you. Oh yeah, found it. All right, so now I'm going to ask Victoria to put people into break rooms so that we can try out different attachment and detachment rates. And this is what we are going to do. Each team will decide on what they want to change on the attachment rate and detachment rate. And then they will discuss what they expect to see. And then run the simulation and see how steady state cross bridge distribution changed and how much and how the force changed, and confirm whether it matches your expectation. And here are some suggested changes that you can make. You can see what is the effect of the lower attachment rate by changing the F parameter here. Or you can also try to make it really sharp by decreasing the W parameter a lot. Or you can make it really wide and see what happens. Or you can actually make the detachment rate go really low on either side by lowering K12 and K22 parameters, or make it go up, or make only one side go down and see what happens. So make some predictions on what you are going to see, and then play with the model and see if cross-bridge distribution and force as a result of it matches your expectations during the steady state and during the shortening and lengthening. And then please share your team's favorite case when we are done with it. Let's take 10 minutes on it. All right, welcome back, everybody. Hope people had fun. Can we now see what kind of predictions people had and what kind of observations they had, and whether it makes sense compared to their predictions? Yeah.
Yeah. We can maybe start with room 2. A couple of our rooms ended up being empty. So room 1's empty. Room 2 that had Amelia, Casey, Kisa, your room.
Perfect. So I said I would speak for our room. So we started with basically just a couple basic predictions. So we thought that if we increased the attachment rate, so I think that's that F parameter, we expected to see a larger peak force from the cross bridge force production, and we verified it, and that we were right about that. And then when we also predicted that some combination of these K parameters, which are the cooperativity parameters and something else, if we offset them, so if we made the K1s high and the K2s low, then we would see an increase in force. Well, actually, some offset of them would create like a maximum production of force. But we weren't quite sure what combination of those parameters would produce the highest force. So that was something that we just decided to experiment with. That's what our group did.
Oh, thank you for sharing. I would guess that maybe that also depends on what kind of lengthening or shortening you do. So a combination of K1, 2, 2, 1, and those things will make your detachment curve skewed. And then if you are going towards that way and then make the curve lower at that point, close splits will be less likely to lose their binding and will be able to produce larger force. But if you pull it the other way around, you may actually lose more close splits.
Right. And that was something that we probably should have changed a little bit more. We kept the protocol as like a lengthening cycle the whole time. So we probably should have switched it to a shortening cycle to see what those parameters would lead to.
Cool, thank you. All right, can you hear from the next group?
Right, so next group would be room 4. So Andrew Thornton, Maddie Chastain, your group. So I have Katrina, Aaron, and Mane as well.
I can talk for us. So we kind of just like explored a little with it all. So one of us like put all of them the parameters at the highest setting, kind of broke the program a little bit, and then at the lowest setting and broke it again. And then in the middle, and we noticed that it was like uh it had like I did the PCA. I put all the parameters in the middle, and then I put the PCA at the lowest. And I noticed that it was like a straight line, and then it plateaued for, I believe, the hill, no, the hill coefficient. I think that was what it was. And then for the cross pitch cycles, it was just uh like a box square, where for the if you put it at like a higher PCA or like a higher calcium concentration, was more of a irregular shape, and you saw more like different, like how you saw it in like when you like just hit with the regular like control. So that was interesting to see at the different PCA levels.
Oh, yeah, thank you for sharing. I think it is also experimentally known that the short range stiffness property, which is the sudden increase in force if you stretch the muscle really suddenly, the effect is more prominent on in the submaximal activation level. So your activation is too low or too high, you don't actually see it too much, but there's some interesting point in the middle. And probably that is where actual muscles are operating in most of the cases. So yeah, that's really fun thing to observe. All right, can you hear from the next group?
Right. I think USC messaged me. They had some errors with their code. So we might have to work on that separately. UCI watch party next. Okay. And then mine's doing late. The Emory watch party. Were you guys able to run the code?
Yes. Yes, we mainly kept the parameters the same. The thing we mostly played with was the PCA level and the protocol in the shortened and lengthened states. And we observed that when it was in the shortened protocol, the force produced was less than the lengthened protocol when PCA was both high and low, right? Yes. Like Hansol was saying, it depends on the state.
All right, thank you.
It's CHP 233D. I have no idea which watch party you are. But if you have anything to share.
Hi, sorry, I guess we couldn't change the name of the room. So we just, can you hear me okay? Okay, perfect. So we just made the detachment rate to be 0 completely and ran the simulation for shortening as well as lengthening. So I guess what we found was even though, so basically detachment rate is 0, the distribution of bond was more or less the same. For shortening, the force reduced, and for lengthening, the force increased. So we thought it mostly remained the same. Maybe it is due to the passive elements, or we're just wondering, are we missing something there?
Sorry, I didn't hear the first part really clearly, but did you say that you put everything 0 and still saw some force developing?
Yeah.
You're right, there are some passive elements. So yeah, even without the close bridge, it will generate some force.
Okay, that's for lengthening, but why is the force decreasing with shortening? Even though the number of attachment remains the same?
That I don't really know. I will need to look into it.
Okay.
But it can be at some weird interaction with the passive elements again. But yeah, I think the way, well, it was actually Tim implemented those elements. So I will blame Tim if it is wrong, but I think it is set such a way that even during the resting, uh, the passive elements are under some tension.
I see.
So shortening would still do something, but yeah, sorry, I'm not super clear about it. So thank you, and I didn't know that, so I will look into that.
Sure. Thanks.
Mhm.
All right. That's the last of our groups.
Cool. All right. I hope people had fun simulating our muscle models. So let me briefly talk about other features of our model. So in the beginning, I told people that there is force length and force velocity relationship in the muscles, and our model is able to simulate that. So for conventional hill-time muscle models, usually those muscle models will start with a prescribed force length and force velocity curve, and then multiply those together to estimate the force at a certain length and velocity. But for our model, when you simulate the model at a different length or different velocity, you can see how much force is developed and then plot it over length and velocity to get this typical force length and velocity curve. So I will show how to do this. Right. One more screen. switching. Where did it go? Share. So many things open. All right. Yeah, here's our program again, and people can try that on their laptop if they like. You can see that actually there was a hill curve on top of the blue curve here on the force curve. And that one I already had some default values for the hill time muscle model for this my default parameters. And you can see that hill time muscle model estimates the force kind of similar to the cross-bridge model. So overall, it simulates a similar amount of a force, but the transient is where the difference is. But when you start changing the muscle properties, let me just reduce the attachment rate because that is the most straightforward thing to see. So now the probability of cross-bridge being attached is reduced. And because of that, we get overall less force. But the hill time muscle model didn't update yet, so it is not estimating the force correctly. So now we can generate the hill type curve using these new parameters by clicking Generate Hill button. So when you click it, what it does is that it will simulate the cross-bridge force at multiple muscle lengths and simulate the isometric force. So you are seeing the force on the graph here. And then also it will simulate the force velocity curve by pulling the muscle at different velocities. But we will evaluate the force when each of the muscles are going through the same length. So for the zero velocity, we will look at the isometric force. For the lengthening cycle, we will start with a slightly shorter muscle and pull it at a constant velocity and see what is the force when it comes to the same length. So by running multiple simulations at different lengths, velocity, and PCA level, these are the curves we get for length, velocity, and PCA curves. I think those spikes happens because of having too low attachment rate, and the numerical methods sometimes becomes a little bit weird at too low or too high values. But yeah, that is how we can generate the hill type curve or the force length, force velocity curves out of our cross-bridge muscle model. And now you can see that hill type muscle model predicts similar amount of a force as our cross-bridge model. And now we have this new hill type muscle model. We can simulate different types of protocols and compare how hill time muscle model and cross-bridge muscle model estimates muscle force differently. I would say overall they are similar, but depending on your question that you are asking, if this transient matters for you, maybe you need a cross-bridge muscle model rather than using hill time muscle model. But if you are looking for, say, average force at steady state, maybe hill time muscle model will be enough for you. Any questions on this? Can people try their own hill time muscle models? I will give you maybe a couple of minutes. And please put thumbs up when you're done with it.
Hansol, I have a question.
Okay.
So haven't people tried to modify this hill type muscle model, like add some history dependent property like length rate or sometimes some kind of additional elements, because I think people have access to a lot of experimental data, right? Why can't they improvise? I mean, have people looked at that already or not?
Haha, that is actually coming next in my presentation. So yeah, there has been some attempts to incorporate history dependence into hill time muscle model. but so far they are limited for different reasons. So I will talk about that a little more later. But the main problem is generalizability. So the way they prescribe the short range stiffness needs to be dependent on what kind of movement happened before, or how much movement is going to happen during your stretches and things like that. I will tell you more with the example later though. All right, can I get thumbs up if people were able to generate their real-time muscle model? Cool. Right, thanks. Right, stop share. I'm going back to my presentation again. Right, so yeah, that was the basics of how our muscle model works. And let me tell you more about what it means for our understanding of the biological movements. So I will tell you a little more about what I'm doing with these muscle models, and hopefully they will answer some of the questions you may have, like what I just got. So, when we look at people's response when we perturb them on a perturbation platform like this, their joint torque are specific to the joint. So typically we see much faster increase on the joint torque on the hip joint, and then it'll go down sooner. And then when the platform decelerates, we see the dip to the opposite side. And then it is a little more smooth in the knee. And then on the ankle, the force develops much more slowly and then stays on the same side, and it doesn't quite go back to the other side. So if we want to use a muscle model to study the mechanism and want to answer questions like why different joints have different torque properties, we may need a muscle model. But let me just show you that real-time muscle model is not quite suitable for answering these questions. This is another data from the human experiment. And here is what happens with the conventional real-time muscle model during the 100 milliseconds after the perturbation. So 100 milliseconds is quite short. It is shorter than time that is typically needed for the feedback pathway to generate torque because of the delay in the neuromuscular system. And on the left is what we measured from humans. And you see very little movement, right? It barely moves. And on the middle is the simulation result using conventional real-time muscle model. And then you see that it overestimates the initial movement by a large amount. And if we get that much of a difference in the initial movement, that may also lead us to a misinterpretation of how much collective torque is needed later to collect this position error. And the previous authors, De et al., showed that they can prescribe short-range stiffness, like the question I just got. They get much better result. So the simulation result looks more like the experimental result. So showing that it is actually needed to have the short range stiffness in your muscle properties. But how they did it was by prescribing the short range stiffness. So they said, well, I'm slightly unclear whether it was time dependent or length dependent, But for this type of simulation, they should mean actually the same thing. So during the initial stretch, they said have higher torque or higher stiffness on your joint, on your muscles. But the problem of doing so was that it was not generalizable. So when they test the model against a different direction of the perturbation, or when they perturb the model by a different amplitude, the amount of short-range stiffness they needed to reproduce human data was different. Meaning, if they fix one type of short-range stiffness, it wouldn't generate to across multiple experimental conditions. And if the short-range stiffness emerges from the muscle model we have that is based on the physiological process of the muscle, we are hoping that it is more generalizable, and thus we can use it to simulate balance response better than real-time muscle models. Did it now answer your question? Was it Sriram, by the way?
Yes. Thanks.
Yeah. Oh. Any questions about short-range stiffness? All right, moving on. So the GUI you played with were showing the case where we impose the movement. That is similar to how we do the experiment on the muscle fibers or muscle in the lab. When you have isolated muscle and you pull it to the length you desire. So the length was imposed to the muscle, but the other way of running cross-bridge simulation is by coupling it with other dynamic system like this. And then the simulation need to meet the constraint that the overall length change of the muscle on both sides should match the change in the angle of this inverted pendulum. And again, there is cross-bridge muscle mechanics inside this muscle model. And we can apply perturbation and come up with a control loop like this. There is the dynamics of the segment. Here it is the inverted pendulum. And then neuron and muscles are connected by the length constraint. And then we can now run optimization studies on asking things like, what is the optimal excitation pattern, or what is the optimal balancing strategy if you have a system like this? By applying the different amount of calcium as a control signal to the muscle. And we can also test the effect of variable tendon compliance by connecting our cross-bridge muscle model to tendons of different compliances. Because it is fun to look at, let me just show some of my results. So this is how much force is developed when you simulate your muscle like what we just did. But if we simulate the same muscle model with a more compliant tendon attached to it, even when the overall length change to the muscle tendon unit is the same, we can see the blue bar moving together for these two muscles. Because when you have a compliant tendon will absorb some of the displacement that is imposed from outside. The contractile element shown in pink here is experiencing much less length change compared to the stiff tendon. And you can run the cross-bridge simulation inside the muscle to see how much force is developed. And because the compliant tendon will absorb the displacement and the muscle fiber doesn't go through a lot of length change, the amount of stiffness it develops is reduced when you have more compliant tendon. And if you put that back into the inverted pendulum dynamics, this is what happens with our simulation. So because the hip muscles are connected to much more stiff tendon, our simulation will show that, okay, the joint torque that is connected to the more stiff tendon will increase much more sharply, where joint torque connected to a compliant tendon will increase really slowly like this. And then we can do the optimization on the feedback gain to simulate the overall control response. And our simulation captures most of the key trends seen in human data across different joints like this. So actually that is end of my presentation, and I will show the sign in link again in case some people missed it. And feel free to stay for more discussions or feel free to leave if you need to go. And thank you for joining today. Oh, and by the way, there is part 2 in the code you downloaded from the GitHub. And if you are interested in simulating something like this, like the inverted pendulum, there is the code in the part 2 custom, if you want to try it. And let me know if you have any questions about it.
Thanks so much, Hansol.
Thank you.
I know we had a few groups too that had some trouble with running the code. This would be a good chance to ask any questions about any of the errors you might have received.
Yeah, was it that you didn't have MATLAB, or did the code run error? Can I learn more about it?
Hello.
Hello.
Hi. Yeah, we were having trouble running the code, but we figured out that it was the way it was saving to our computers. I guess we had to change the folder, like the place it was saved for it to run. But we did try to get it to run, so the GUI popped up. Yeah. Thank you.
All right. Yeah, I think you will need to be in the folder and probably maybe try to add other subfolders to the MATLAB path, if that helps.
I wonder if it needs to be out of the downloads folder.
Like,
Yeah.
oh,
sorry.
Oh,
I
sorry.
had to zip the folder first and then like export it and then move it around. Yeah.
MATLAB is very fussy about where things are. So that is always the first thing to check when your code doesn't run. Hansol, I had a more of a science question, maybe while other people think about coding questions. And that is, so you had the nice prediction from your model that tendon compliance could be responsible for the different responses at the different joints to perturbations. But have you gone back to the literature to see whether that hypothesis is supported by data on the compliance of the different tendons at the different joints?
I've looked at some, tendon compliance from the literature, and so far it seems like so. Like Achilles tendon is really compliant, and the tendons on the joint around the knee are little less compliant, but still kind of compliant. Where it seems like the muscles connected to hip joints are most stiff. But I looked at a few muscles that are around the joints. Seems like so far there is some gradient across those joints. And I wonder whether it actually means something for the function of the joints, proximal to distal. And I think there also is a suggestion that Achilles tendon stiffness is maybe optimal for the frequency of our walking and running, for the optimal energy return. So it might be compliant tendon might be a feature that is helpful for the energy, but maybe hurts a little bit in terms of balance if it slows down the resistive torque development. Do you have any thoughts?
Well, I was just thinking that just all other things equal, which of course they aren't. Of course, the tendons are longer in the distal limb, and of course, if you have just a longer spring with the same properties, it's going to be more compliant. So it partly could be like there could be like this anatomical constraint that actually is somewhat adaptive as well. Does that make sense?
Yes.
Yeah. 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 Nice work.
Thank you. Any other science questions or anything?
I had a question. So I'm very new to all of this, and so one of the questions that I had was I'm currently working on very experimental work, so I don't really understand too much about the computational. But how do you come up with the numbers for the attachment rate and detachment rate and the number of attached cross bridges if you can't experimentally see them?
Okay. That part was largely done by my colleagues Tim van der Zee and Solveig Sima. But I know that some of those comes from the experiment. So you can measure what is the like natural attachment and detachment rates. I think some of them actually you can track to the molecular process. And some of the rates, they fit the model to a single fiber experimental data.
Oh, okay.
So yeah, fit the data to the single fiber data and then use that model to predict the behavioral data later.
Okay, that makes sense. Thank you.
Yeah, I think some of those parameters are somewhat bound to physics or chemistry, where some of those are more of an effect of an overall muscle structure and is different across species or different across muscle types. So I think Tim and Solveig did a good work of looking into the literatures to see, what are numbers they can just grab from some physics parameters or physics what you call this constants. And some of them they fit it, to the single fiber experiment.
Okay, thank you.
Hi, Hansol. Very nice talk. I was just wondering, what are the next steps for this model? Are you guys thinking about implementing in a multi-muscle, like a lower extremity task? And then the second question is, how intensive is the computations? Can we how do you think it's going to pan out? Yeah, I'm just curious.
All right, thanks for the questions. For the model next step, there are multiple ways we can go. One of them would be doing maybe double inverted pendulum rather than doing single inverted pendulum, because obviously those lower limb joints interact with each other. That is one thing. Other thing, we can extend the model to have a more realistic energy model, because this model is based on the molecular process within the muscle, and we know like how much ATP is needed for cross bridge to bind and things like that. Probably we can have a better model of energy of the muscle, and maybe can use that to run the optimization on the trade-offs in biological controls. I am interested in the control aspect of it. So I'm making the feedback loops and optimizing it and seeing how it changes when there are changes in the system, like when you have some disease condition and have increased delay, increased noise, or maybe some wiring between the sensor loops get a bit altered. What is the effect of it, and what are the adaptation strategies given those conditions? What was the second question? Sorry. Computational time?
Yeah, so if suppose if you're using it in a like a whole body simulation, can we replace it with the hill type muscle model and do computations, do forward simulations, and so on? Like, are you guys thinking along that lines?
I don't have an exact number at hand right now, but closed simulation is computationally more expensive than the hill type muscle model. That is for sure. So there has been some efforts made to make it more efficient. So rather than say keep track of the overall distribution of the bound crossbreeds, Tim and Friedel at KU Leuven are working on the right, looking at the name, but direct moment model. So by approximating the cross-bridge distribution to a Gaussian distribution, they can come up with a mathematical equation of the like moment of the distribution, first moment, second moment of the distribution to calculate the force more easily, and then they can simplify the transition equation by doing so. So they can calculate the transition of the overall Gaussian distribution into another Gaussian distribution rather than evaluating transition at each point on the distribution. And by doing so, they were able to make it more computationally efficient. So from my experience of working with the model, it seems like running the direct methods, so without the approximation of the distribution, I think close splits takes like 10 times longer than the hill time muscle model for my simulation. And with the approximation model, it seems like it is around like twice or three times longer. So I would say it is not crazy longer. It is not like 100 or 1,000 times longer, but it is a bit heavier. But it's getting better. And Tim and Friedel are also working on making the approximation model to work with the OpenSim, so they can import their version of the approximation cross-bridge model into the OpenSim to run the full body simulation.
This is great. Thank you.
Okay.
I had a question, Dr. Ryu.
Hey, you can just
So
call me answer. Yeah.
yeah, so I'm Mane from Northern Arizona University. Could
Mhm.
you go to slide 12 of your presentation?
Sorry, which one?
Slide 12, I think, is the one.
Great. Right now.
I was just, I didn't fully understand what the distribution of bound cross bridges means. So I was wondering if you could explain that again.
Okay, sure.
Yeah,
Maybe something like this. Yeah.
yeah, exactly. So you got that plot there, the third one from the top, percentage cross bridge bound. Um, and the x-axis is delta x, so that's the length change of the cross bridge.
Right. So, yep, that's right. The close splits is at various different lengths
Yeah.
compared to their resting lengths. And then some of them are attached and some of them are detached. And we can look at the distribution and count how many close splits are within certain certain displacement
Okay.
from the resting lengths and see how many of them are attached at that length. So we will discretize the length. So this one will be say at length 5 and this one is at length 0. And we count 1 and 1 here. And then look for the whole distribution and look for the like overall distribution of the bound cross bridge. Did that help?
Yeah, I think so. So the if that distribution is centered at 0,
Right.
that means that on average, all the bound cross bridges are at equilibrium.
Right. We have highest number of cross splits that are bound at the center or at the equilibrium. And
Okay.
that is because our attachment rate is highest at the equilibrium. And that kind of makes sense because it'll be easier to bind if they are, aligned.
Yeah.
And then it becomes easy to lose it when they are pulled too far away.
Okay. So when the distribution is centered at 0, you can still have force being generated.
Right.
But what does that mean for, say, the shortening of the entire fiber? Does it like, to me it seems like if it's centered at 0, the fiber is not going to shorten. Is that correct? It'll just be at its equilibrium length.
So there are actually two types of forces going on here. One is the spring-like force. And another one is the cross splits cycling force. So cross splits goes through the power cycle like this. So it actually actively pulls the filament. So you will still get that force even if your forces are centered around 0. So even if your displacements are centered around 0. And I
Oh,
think
sorry.
you're right that, if we only look at the spring-like force,